well, super exciting for us, and it's where we're going to go next. And this is the bit where you guys can get involved too. Yeah, so yeah, so just if you have heard me talk recently, so I gave a talk in November time, but I just want to say that there is lots of new data coming in here. So a lot of things have happened since I last spoke. So there is a reason to be here. So thank you for turning up. Okay, so part one, we're going to add some extra awesomeness to an already super family. And this is a story about laminin alpha 3 and alternative splicing, really. So before I sort of launch into the laminins, I just want to make sure we put everything in context. You probably learned about basement membranes when you were in school. They're the point that sheets of epithelial, endothelial, nerves and muscles attach to that provide that barrier layer between the sheets of cells and their underlying matrix. And they are ubiquitous, they are everywhere, and they do lots and lots of things. Now, the sort of fundamental thing, the thing that they're best known for is this, for the attachment site, for providing that support for cell adhesion and tissue integrity. And this disease here is a disease called junctional epidermolysis bullosa, where mutations to any of the components of the basement membrane or the adhesion complexes lead to this skin fragility disorder. And the bottom right are two variants of that, where it's autoantibodies that target basement membrane proteins. And the way that tissues are held together, so obviously this is the skin, but this holds true for other tissues as well, are via specialized adhesive devices. And this is an electron micrograph of a hemidesmosome, which is the skin-specific kind of spot weld that holds the skin down. So this has been understood for, I don't know, 30 odd years, that the basement membrane is key for holding your tissues together. But they do more than that, of course. So the same proteins that are required for stable attachment are also, they provide the substrate for cells to migrate. This is an example of cell migration. So this is a wound that's just healed in a back of a rat. And the purple in here is one of the major basement membrane proteins that's being upregulated in the wound bed. And that's part of the whole process that the cells will bind to and migrate over. Migration obviously happens in lots of contexts, and the biggest ones being development, and I'm going to show you pictures from our mouse later on where we're looking at the developmental stages, and also all tissue morphogenesis later on. So whenever there's a change in the tissue, such as the hair follicle cycling, then you're seeing a change in the basement membrane structure. So these are hair follicles, by the way. Okay, so the same things, the same proteins are adhesive and support migration, but it's also a barrier too. So the basement membrane is a selective barrier. It allows certain things to pass, certain molecules and certain cells to pass. So it's required, and holes are made in it to allow immune cell extravasation, i.e. leaving the blood vessel and into the stroma. But it also provides a barrier that prevents tumor cells from invading into it. And so one of the hand-to-hand stages of cancer progression is breakdown of the basement membrane. And it's the difference between being metastatic or being benign. And I'll show you some cancer data later on. But then on top of all that, so it's already doing important structural things, but what's kind of become the big deal in the whole basement membrane field is that it's actually a massive hub for signaling in lots of different ways. So the proteins themselves provide a ligand for cell surface receptors to bind and initiate a whole bunch of signaling cascades, but also the structure itself sequesters growth factors, and those are selectively released to create gradients, for example, in a wound repair context, that can influence the cells in either direction. And things like the end of the fibrotic response. So in a wound environment, you have your fibroblasts becoming activated, producing fibrotic material. It's only once the basement membrane reforms and you block that signal coming from the keratinocytes down to the fibroblasts that the fibrotic response stops. So there's a whole bunch of things that the basement membrane is super critical for. And the big questions are, how does it do it? So to talk about that, I need to tell you what's in it. So basically every basement membrane is based around 4 components. Laminins come around first, and then they're bound by nidogens, first of all, and then later on perlicans. And they provide a hub to which the collagen 4 network can assemble. So every basement membrane has these 4 components and then a whole host of other things as well. This diagram, which obviously is beautifully drawn, is actually fundamentally based on what it actually looks like. So this is a really old paper, well, it was a 1992 paper, where the EHS, so this is the same stuff that Matrigel is made from. The mouse sarcoma basement membrane type material has been selectively degraded So the collagen 4 has been degraded and you see the hexagonal array of laminins, or if the laminin gets degraded, you see the collagen network. So it's a real structural entity. And when you have a real structural entity, sometimes, oh, it's not playing. Oh, that's the problem with this pointer. Yeah, when you have a structural entity like this, you can get your students to build a pipe cleaner model of it. So the vertical struts here are the laminins. Along the top here, you've got the collagen 4 networks, and then you've got these basically laminin linkages. And if you're ever on the second floor of the William Henry Duncan building, you can see this model and you can press on it. You can see that this is a structural entity. Okay, all good. All happy so far. Okay, so I'm going to talk about laminins. And I just want to point out 3 of the major structural features of laminins. At their C terminus, down here, we've got the laminin globular domains. And they provide most of the high affinity receptor binding sites. So you're the binding sites for your integrins, your dystroglycan, and syndicins. Most of the highest ones are in here. There are other binding sites, but these are where most of the sort of, where the cells hook on, if you like. Over here, we've got a laminin coil domain. This is the multicolored stripy bit. And this is where an alpha chain, a beta chain, and a gamma chain form together to form this heterotrimer that then gets deposited into the cell. So every laminin molecule, by definition, is an alpha, beta, gamma heterotrimer. Whereas the 3 colored blobs, the yellow, blue, and red blobs, are called laminin N-terminal domains, LN domains, and they provide the site for laminin to laminin interaction. So the formation of that network that I was showing you earlier happens up here. All good so far. But I told you that there was a missing member of the family. And to tell you there's something missing, I have to tell you what was there. All right. So if we go back far enough, then you go back, you get to the stage where you only have 1 laminin. Over the course of a long period of time, we get gene duplication events. And that's like most matrix proteins, laminins are modular. So we have the same structures. So through gene duplication, then domain reorganization, very quickly we ended up with multiple alpha chains, a gamma chain, a beta chain, and then one of these, which is related to the beta chain, but it's a separate gene family that contain only the LN domain and a stretch of LE repeats. And this is kind of like a beta-like fragment. More gene duplication events, and then suddenly we have a separate gene producing a gamma-like LN domain fragment. We duplicate a bit more, and we get another beta-like LN domain fragment, and this one's an imperfect LN domain. It has one part that works perfectly and one part that doesn't. We do some more gene duplication events, and eventually we get to what we're close to being now in humans, where we have 12 genes, 13 major splice isoforms, at least 4, possibly 5 different LN domain fragments. And you notice what's missing. The bit that's missing is the yellow version of these little things. We don't have an LN domain fragment. And that is what we work on. So that's a protein that we now call laminin N-terminus alpha-3-1. And the history behind that is a longer story, and I won't go into it too far today, but basically I was studying one of these rare diseases, and we realized that there were transcripts being produced from this gene, from the laminin alpha-3b gene, that encoded this short protein that we've now gone on and proven exists. And I'm going to show you some data that shows that it does stuff. Now, since we reported that, or since I reported that in 2009, we've identified a bunch of more. So there's another member of the family, LaNt alpha-5. This LaNt alpha-1 isn't called that really, but this is a proteolytically released fragment that's generated from the alpha-1, and it seems to be functional during embryogenesis. And there's a proteolytic fragment from alpha-3b, which again is been shown to exist in vitro and regulates a whole bunch of things. So I feel like we've got it now. We've got the collection that you're looking for. You've got these short fragments being produced from each of the different things. So why is that relevant? Why could that be important? And to tell you that, I need to show you the rest of the sort of story of how networks form. So I've said already that they bind via these LN domains, but I just want to give it a little bit more nuance. And so the way this actually works is that you have a beta LN domain, a gamma LN domain, and an alpha LN domain. They all need to come together to give you a stable complex. And the reason that happens is you need, first of all, the beta gamma LN domains to interact, which is a rapid interaction and an unstable interaction. And it's only once you've added the alpha chain to the process that you actually get the stable ternary node forming, the 3 LN domains together. All good so far. Okay, but the thing is that you mightn't have noticed when I was showing you the evolution is that some of those domains disappeared along the way. You had different laminins with or without the LN domains. And what you've effectively got now is 4 different classes, if you will, of laminins. The archetypal, the kind of the classic, the kind of evolutionary ancient version, which has 3 LN domains, 1 on the alpha, 1 on the beta, 1 on the gamma, and that would form the network perfectly. But then in places like blood vessels or in the alveolar epithelium, the laminins that are expressed there only have 2 LN domains, whereas those expressed in the skin only have 1, and this one's quite rare, and whether it exists or not is questionable. Once you layer on top, now you've got these 3 fragments that exist that contain only the bits that are missing. You could imagine that, and this one certainly does, the netrin 4 will compete for the binding sites in here and disrupt the ternary node formation, whereas the alpha chain perhaps can compensate for the missing one here, which would compete again over here. So we've suddenly got this potential to titrate what's going on in the cell, titrate the level of network assembly that's going on. So that's the sort of global picture. Now let me show you some data that supports that global picture. So I'm going to talk about LaNt alpha 3.1 from now on. As I said, it contains an alpha LN domain. These blobs here are laminin type EGF-like repeats, and various people have shown over the years that in this context, that's probably important for protein stability rather than any functional context. It has a unique C terminus. So this is produced by intron retention alternative polydenylation, which encodes for 54 unique amino acids, which is why we've studied it actually, because it provides us with a point in which we can raise antibodies. And we've raised antibodies with mouse monoclonal, rabbit polyclonals against human and against the mouse forms. And just to show you some of the validation of those antibodies, because I'm going to show you some images, we can show that our protein recognized with high specificity, the LaNt protein, and this is when we've overexpressed it in cells. It's actually quite sensitive. This is a dilution series of the purified protein, and it doesn't recognize any of the other laminins, which is what we'd expected because it's a non-conserved region anyway, but this is an ELISA to show it's recognizes the right thing only. On top of that, this is a rabbit polyclonal compared with a mouse monoclonal when we look at oral tissue, and we see a very similar distribution pattern. So both antibodies giving us the same things. They're raised in different ways against different parts of the unique region, makes us even more confident that we're seeing the right thing. And what you see is expression along the basal aspect of the epithelium and around vessels. This is larynx, I think. So a couple of years ago now, we published the distribution in the front of the eye, in the ocular surface. And what we see is there's light expression in the cornea and the conj, but it seems to be enriched in the limbus. And again, this is a comparison between our mouse and our rabbit antibodies showing you that we get the same thing. When you look by fluorescent microscopy in relation to laminin 332, the major epithelial laminin that's produced, we see that the LaNt is enriched just apical to where the laminin is. So it's at the basal aspect of the cells and these upward things. So that's stuff you guys have seen before. Let me show you some new stuff. So I'm going to show you later on some oral squamous cell carcinoma. So this is a normal larynx to show that as you'd expect in epithelium, we're getting something a different type of epithelium. We're getting something very similar to what you see in the eye. I showed you already that it was expressed in vessels, but that's true across a wide variety of different tissues. In the breast, what we see is a terminal ductal lobular unit. We see the basement membrane lighting up, and around these outside the smooth muscle ring, we see LaNt being enriched. And very similarly in the lung, what you have are these linear rays below the surface epithelium, below the alveolar epithelium. And again, I'm going to show you some more lung images later. I'm also going to show you the kidney. You've already seen it if you were here early. So in the kidney, you get LaNt being expressed. You have it around the glomerular basement membrane, and you have all these different tubular structures with variable levels of intensity. And since we did this, there's actually been another paper come out to show that the laminin alpha 3 gene is active in this point. So this is actually fits quite nicely. I will show you some liver pictures at the end. In our liver, we're not seeing any really specific distribution. So, but perhaps someone could help me interpret that a bit. And then I'm not going to show too much more because we actually process pretty like a lot of tissues, and I could give you a whole talk on just where the protein is, which is interesting, but doesn't actually tell you it's functional. So just to say, if you want to go over these sorts of things with me, if there's a certain tissue that you think might be interesting, then I'd be certainly happy to do so. And we have, as I said, we've got quite a lot of data, because it's really important to us to know where the protein is, because that can tell us where it functions. Because on the back of understanding where it is, we then can layer on which laminins are expressed and begin to predict about what it's doing in different cases. I just want to show one or two other examples of things that we didn't expect but then observed. So this is in the cerebellum. I know that Paul's on it, so at least somebody will be keen, probably Richard Barajolli as well. We have Purkinje neurons and axonal type structures being lighting up with our antibody quite specifically. And then also when you look in the retina, which is an extension of the brain, of course, what looks like the Muller glia have got a very definitive staining pattern. So we've got these two enrichments. And just to say, this was all human tissue. We're beginning to do the same thing with our mouse, or Connor's beginning to do our mouse type studies. When we look in the epithelium of the mouse, we also see pretty limited expression in the actual epithelial tissues. You see it around the hair follicle down here in the basement membrane-like structure, but the vessels light up really strongly. And when we do a serial section and then stain with CD31, you can see that we have the large vessels are stained with the LaNt, but it seems that the smaller vessels are less enriched. So we have differential distribution of this protein across the different maturation of the vessels. Okay, so the protein's expressed, and that's interesting, and that's nice. Yes, in terms of the endometrium, I'll send you the pictures separately. Okay, so we know it's expressed, but that doesn't mean it's functional necessarily. So let's look at some situations where we're looking for basement membrane remodeling to be happening. Okay, so I've shown this before as well to some people. So this is an alkali burn model we did with an ex vivo corneal wound. So this is a pig cornea that we've kept in culture, then burned it, and then stopped at various stages. And what you can see is that the LaNt protein is upregulated during the early phases of wound repair, but then returns to basal distribution once the epithelium has healed and is beginning to mature. So we lose the specificity of direction and possibly turn it up. In the limbus, which remember is a region that we saw it was enriched in unwounded tissue, we actually see a much more dramatic increase. It's like it's being turned on in the limbus. Now, the corneal limbus is the point that provides the epithelial cells that become the central cornea. So the limbus is either side. And that's where the sort of stem cell niche is. And so again, we've published this paper recently as well, that we used explants. So this is the corneal rim that we put into culture to activate the stem cells, activate if you like. And what we can see is in the activated explant, so this is the tissue growing down the side of the little chunks of tissue we've grown, we see the LaNt protein being increased as it grows. So we think in stem cell activation, we get an upregulation of the LaNt expression. It's when you hit the cancer, things go a bit crazy, which is no surprise. Lots of things go crazy in cancer, especially when you're talking about alternate splice isoforms. But what's really striking is in the normal epithelium, you've got this basal cell expression that I've shown you already. But as you move in towards from dysplastic into invasive squamous cell carcinoma, this is a tongue carcinoma, you see this dramatic increase in expression. And so obviously one slide doesn't prove anything. So what we did was we processed 90 other oral SECs and advancing fronts, and then scored them based on the relative intensity of their LaNt staining. And what we see is, I need to put the key on. So the ones that actually have the lowest LaNt expression were more likely to have nodal involvement and extracapsular spread. Those two things are obviously linked to each other. Also, and this was somewhat surprising, the ones where, well, all things go together, of course, those with the lowest expression actually have slightly better survival outcomes, though note the p-value is not reaching statistical significance. And again, the ones with the lowest expression were more likely to have recurrence. So it seems, at least in this context, that having high LaNt expression is in some way protective against various things, against nodal involvement, ECS, and recurrence, and perhaps against survival. We also looked in breast cancer, and again, this is normal tissue. These are just 4 examples of normal tissue, 4 invasive ductal carcinomas, 4 invasive ductal carcinomas that are also triple negative, and we see that the LaNt protein was highly expressed. So again, we processed a lot of these. So this is 25 matched samples where we have the uninvolved versus the tumor, and we're seeing that we have an increase in LaNt staining intensity in about 50% of the tumors, whereas when we look in the tumor, against the matched person's nodal tissue, we see this further increase. So it seems that LaNT's upregulated in cancer and then upregulated again in the nodal metastasis. But despite that, that seems like a really nice finding. However, when we looked at other aspects, it doesn't seem to be particularly predictive for outcome. Or this isn't necessarily all outcome. So top left, tumor grade, in the highly LaNT expressing cells, we have slightly more of the higher grade, but it's nowhere close to being statistically significant. There's no effect on nodal involvement, and then survival in this particular cohort's not, most of them survive anyway, but there's no difference between the two. So it's not necessarily predictive of outcome, but certainly it's associated with the cancer itself. Okay, so far so good. The N here, we're into the 150s. Oh yeah, just one extra thing. When we look for triple negative in particular, so these are estrogen receptor negative, progesterone receptor negative, and HER2 negative, 2/3 of them have medium or high expression. So even though it may not predict for outcome, it could actually become a drug target that's specific for these triple negatives that don't have other drug targets. But the bigger question is, does it actually do anything? So if we go on to my third part, which is beginning to look at what LaNT as a protein does, and I'm going to run out of time if I go at this speed. So let me probably rush through this bit. So we saw no effect on cell proliferation. What we did see was a decrease in wound closure rate. We also saw a decrease in cell migration speed. So the individual cells move less fast. And this is across a bunch of different epithelial cell types. The cells spread further. They had an increased two-dimensional spread area, and they had an increased adhesion strength. I'm sorry I'm not giving you time to sort of absorb the data here, but I'm happy to go through this separately. So at this point, these are a bunch of phenotypes that are all like important, but they don't necessarily say it's the cells or the ECM. And so to ask that question, we've done cross-matrix assays where we've taken the cells, allow them to elaborate a matrix, remove the cells, and then put fresh cells on top, and then ask the same questions. Does the matrix rescue the phenotype? And if we do that with an ECM for the cell migration, yes, it can be rescued by the ECM. And if we do it for the two-dimensional migration, two-dimensional cell area, that also rescued by ECM. We also obviously are interested in cancer, which means that we're interested in cell invasion through a matrix. And so for those experiments, we did this experiment where we plated our invasive breast cancer cells on the bottom and provided a matrix, and then the cells would invade through it, and we can measure how far through they invade. Now, so we asked the question, okay, if we put a collagen type 1 matrix in place, do the cells invade more rapidly? The answer is nope. No, they don't. So no extra invasion into collagen, less invasion, doesn't seem to affect their invasion into collagen in any way. But when we did it into Matrigel, and Matrigel contains predominantly, well, 60% laminin 111, about 30% collagen 4, and then thousands of other proteins. But what we do see there is actually a significant reduction in the total depth of the number of cells per area. So we get a slight reduction in invasion. What was perhaps more striking is that when normal MDA MB231 cells invade into collagen, they do so with this multicellular streaming that kind of go together as a group. Whereas if you do the same when we have overexpressed the LaNT protein, it's individual cells that invade instead. So you can see that with a maximum intensity projection, so that in this Matrigel, so this laminin specific phenotype, we get multicellular streaming normally, but we lose that. So when we have excess LaNT, so we lose this cohesion of invasion. And then when we go back and look at the tumor sections, and look at the relative level of cohesiveness in relation to how the highly expressing LaNT tissues, and we see that you could classify the high LaNT expressing ones as non-cohesive in about 70% of cases. So it does correlate with the phenotype, but it doesn't necessarily mean we're directly seeing that. So, okay, so together that says we've got changing the mode of invasion into laminin-containing matrix. So all this is pointing to a laminin or matrix effect. But when we look at the effect of overexpression on the laminin expression, there isn't any. At least there's not anything major in terms of absolute abundance of the proteins. All the laminins we've tested are expressed at the same level. But we never necessarily expected that. What we did expect is that the LaNT would bind to the laminin and change the way that the laminin was structured. And we do see that. So we see this change from swirls and arcs in the control cells into tight clusters in the LaNT overexpressing cells. And what we also see is that when we express the GFP tagged form of the LaNt and an M-cherry tagged form of laminin beta 3, that there's nice co-distribution between the two signals with total internal reflection microscopy. And we can zoom in on that and you can see that they are quite close. It's not perfect. This is a protein that's associated with the cell as well, and this is deposited very efficiently. So we have an imperfect, but it seems to match. And you can see that again, and perhaps with, it perhaps makes more sense if I show you the live movie, and this is a maximum intensity projection of all the planes together, that the LaNt protein in the middle here distributes nicely on top of the laminin and bounces around the edges. So this part here, you'll see it comes and goes, and the same here, it'll come and go as it goes, and it'll avoid the back. So we're actually seeing them interact. Okay, so very quickly, 3 different sort of biochemical type assays to see whether they can interact directly. Left-hand side's an IP, and we can pull down the LaNt, we pull down laminin beta 3. The middle one, we spiked in the LaNt and pulled it down, and we can pull down laminin alpha 3. And the far right is a far western, where we've probed the western blot with a LaNt protein and then detected the protein, and it matches up to laminin sized band. So we think it can bind, it at least can form a complex. This assay is a bit weak, so we need some tighter experiments to be really confident. And it all makes sense, because when you balance everything, you balance the change in migration against the change in adhesion, that all makes sense, because really these are different sides of the same coin. If you have really strong attachment, your cells are unable to generate the force required to move. And there's some Goldilocks level where you get maximum migration. And what I'm really saying here is the LaNt protein provides that way of titrating between adhesion and migration strength. It provides a switch between the two. And I am going to skip through how it does so, because this gets a bit more complex. Because I want to get to the mouse. I'll skip the chickens, and we'll skip that chicken, and we'll skip that bit too. The real reason I wanted to give a seminar today was begin to show you this part of the project. And this is what's effectively known as LaNt Wars. So, yeah, okay. So basically, maybe I'll show you this. No, okay, fine. I have a conversation with myself, because I figure there are people out there in the void. Anyway, okay, so all the data that I've shown you so far is somewhat kind of correlative. We either have an increase in expression in tissues that is associated with a behavior, or when we modify expression in vitro, we see changes in cell behavior. And what I haven't shown you, or have time to show you there, it does so by changing the cell matrix adhesions and MMP activities. But what we really need is to put things into the true physiological context. And so this part of the talk is 100% new. I've not presented it to anybody anywhere before. We've had a few chats around the place, so I don't think anybody will have seen this apart from George or anyone in my team. It's been a saga. It's been an ongoing thing. And let me start at the beginning. So my first PhD student, Valentina Iorio, who some people will remember, in her final sort of period of her PhD in her last year, we made a mouse where we had a keratin 14 promoter driving the immunoglobulin kappa leader sequence, which is followed by the full-length human LaNt alpha 3-1, a T2A sequence, and an M-cherry. So what this means is you have pretty much epithelial-specific expression of the LaNt protein and the M-cherry, because this T2A element allows 2 proteins to be produced from the same gene, basically. So this gets cleaved here. So it's an untagged protein apart from the histine tag on the end. I don't think that makes sense. So she did all the tests you'd expect. We tested in vitro, made sure we got expression when we expressed the thing in epithelial cells, but not endothelial cells, for example. She did the blots to show it expressed. But when we actually got through to the actual testing transgenics, we had 2 animals that had the transgene, but we could not detect any protein expression in those animals. And that in itself isn't unusual. The transgene can land anywhere when you put it into the animal. What was surprising was how few animals we got back, and the only animals we got were non-expressing. And this, together with a bunch of other things and a bunch of underlying sort of knowledge about what could be going on, made us think that this might be a lethal phenotype, which was thoroughly depressing, because when you don't have the animals in your hand to actually prove that they were it's lethal, you can't be sure about anything, and we couldn't publish anything. And this obviously pushed us back and made us unhappy for a while. However, we got a new student. So this is now, we're on to Connor's work. And we came up with a different plan to allow us to move forward. So this is all done with George, and I can't thank George enough for helping us every step along the way here. So what we've done that's different in this construct, so we've used the ubiquitin C promoter. So that gives us ubiquitous expression. You can guess from the name. So every cell and every tissue should express the transgene. But we've introduced this stop cassette at the front. So this is a flock stop cassette. So these two LOX P sites means that this 3 times poly A site will prevent transcription from happening until such a point as these LOX P sites are removed, and they can be removed by Cree recombinase. I hope that all makes sense. So basically this is inducible now. So when there's Cree is active, we'll chop those out and we can drive the transgene. But if there's no Cree, no expression. And we have the same construct, full length human LaNt with a couple of tags on the end, a T2A element and a TD tomato, which is a slightly brighter form of M. cherry, different fluorescent protein. So Connor then did all the usual, the cloning to make all this, and then he did a power of pre-making the mouse validation. So this is just an in vitro test where we've transfected in either the plasmid he's made or Cree with a GFP tag or both. And you see that when we have the GFP Cree, we get the purple, which is the TD tomato signal. We get the recombination, we get the protein express. So those are the in vitro tests. And then we made the first transgenic line that was carrying this transgene, and he grew mouse embryonic fibroblasts from that transgene. And what you can see is when there's no Cree in these animals, you don't get any protein express. But when you add the Cree in vitro with an adenovirus, we can induce expression. And this is us detecting it with the HA tag, which is one of the tags, whereas mess from wild type cells, even with the Cree, we don't get that. And you can see bottom right, that's these cells again. We get the signal that we're looking for. We get the tomato signal coming from the adeno-Cree. Awesome. So we've got the construct working very nicely in our hands. And next, then, we're in a position where we're able to start asking questions about what this thing does, what happens to the animal when we express it. And so we've crossed it now with a ROSA26 Cree estrogen receptor. So what this means is that the ER holds the Cree at the cell membrane until such a point as tamoxifen is added, at which point the Cree translocates to the nucleus and then can remove that stop site. So basically, once we add tamoxifen, we can turn on expression. The ROSA26 also is expressed everywhere. So basically, our tamoxifen treatment, if it's topical, could be topical, but generally speaking, if we do a systemic treatment, we'll get expression of LaNt everywhere. Testing those animals, because obviously we're going to test every single step along the way, we see exactly what we want. Tamoxifen treated of the heterozygous cells in a dish. These are explants from skin. We can see we can turn on the TD tomato signal and we can turn on the protein. So things can express. Big sigh, because that means we're awesome, we're off, we're ready to ask our question. And one of the things I said at the beginning is that laminin and basement membrane remodeling is a huge deal during development. And so the first questions, the big questions we've asked, the biggest chance of us to see a phenotype is that if we induced expression at embryonic day 14.5. So mice embryogenesis is 20.5, 21 days for them to get born. So we've induced expression at 14.5 and then let them carry on for the remaining 6 or 7 days before the animals are born. And so what happened? Well, they died, which was actually great news. So I know that sounds crazy, but what we had was we've had 2 separate litters, one that was born in December, the other one born in January. The first litter, 2 out of our 5 animals, and the second litter, 3 out of the 6 animals, I think, were born intact but dead. And why that's good is that's exact Mendelian inheritance, what we'd expect from a het mating. And what we have that you can see is, okay, there's no major skin defects, but what we do have is this erythema, this redness of the skin that's likely due to blood vessel leakage. Now, it's in different places than the different dead animals, so this isn't just this particularly got handled on top of the head or anything like that. So the mice are yeah, so that's what happened, and on the gross scale. Obviously, this is now labeled expressing and non-expressing, but that needed to be confirmed. So we have confirmed that with cells in the dish that were explanted from the things, showing that they have the TD tomato being expressed. we have the blot, whereas an explant from a non, from a, an alive animal was fine, whereas the explant from the expressing embryo expresses the protein. And then when we look for the M-cherry, or the TD tomato signal in the skin, using an antibody actually to detect it, to amplify the signal, we can see that we have lots of expression in the expressing animal that we don't see in the non-expressing. So so from now on, you'll see them labeled expressing and non-expressing because we did these tests. And what I want to show you is 4 different tissues. I'm going to show you the skin because this makes a lot of sense because we've studied the skin a lot. But also when you look at it, consider it to be a 1 LN domain containing laminin. Every tissue is a mix, but this is the major laminin that's present. In every tissue, the blood vessels are a bit messed up. So just remember that that's predominantly a 2 LN domain containing basement membrane. I'm going to show you just one picture of the lung. We're not 100% sold on it, but that's also a 2 LN domain containing tissue. And I'm going to finish with the kidney, which is also where I started this talk, which is a 3 LN domain containing laminin. I'd also point out that at E14.5, almost all the tissues are expressing lamin 111, and it's only when they become a bit older that they begin to turn on these other laminins. So they've grown up in a situation where there's 3 LN domains. Okay, back to the skin. Okay, skin, laminin alpha 3 is the laminin 332 is the major one. Just to quickly say, when we were predicting what was going to happen here, I thought they might have junctional epidermolysis bullosa. They might have that skin disease that I showed you right at the beginning. And quite simply, if you knock out laminin alpha 3, you get skin fragility, you get blistering. And I've worked with these animals, they're born dead, but their skin falls off. And this is not what happened here. And I'd also say that I thought we might get a mild version of that phenotype. Now, this is domain 6 is the old name for an LN domain. So these are skin equivalents that have been made where that LN domain's been depleted. And they've shown that you get mild skin fragility and excessive granulation tissue production in those deletion constructs. So those are kind of what we were looking for, or that's what I was expecting if we were right in that an LN domain function would be inhibited. But you can look at our skin H&E and make your own decision, but there's not a huge amount going on. It's not nearly as striking as I expected. Now that sounds depressing. You can see these bright fuchsia kind of dots, those are almost certainly red blood cells. But what you can see is it's much more subtle than I hoped, I guess. The very tight linear association between the epithelium and the stroma seems to be messed up. That point, that dermal epidermal junction, is a lot less kind of defined, if you will, in our expressing animals. The expressing's on the bottom, by the way, and all these images, and the non-expressing on the top. And this is a hair follicle where you've got the same sort of idea. If you look at the laminin expression in these animals, you can see that to a greater extent. So again, the laminin is expressed. This is pan-laminin, so it's mostly laminin 111 we're looking at. And if you zoom in a bit, you can see that the laminin here is well, in the non-expressing animals, you have a pretty definitive line where it's a kind of fuzzier line, but the hair follicle seems to be all right and possibly thickened. Now, what I didn't show you because I zoomed past it was when we overexpress LaNt, we actually recruit collagen 17 more rapidly to beta-4 integrin, 2 proteins I also haven't talked about, so don't worry too much about them. But what I would say is we looked at collagen 17 because of that, and we actually see this rather striking phenotype so this is a hair follicle, and what we have is that collagen 17 now traverses into the neck, this is the bulge region of the hair follicle, which you don't normally see in a wild type animal. You see this light expression 17 and perhaps down the dermal papilla area. So this, we seem to have a mislocalization collagen 17. Anyway, so the skin phenotype wasn't too dramatic. The difference between the skin and the lung, even though they both produce laminin alpha 3, one produces 311 while the other produces 332. And this is an old image that I took of using the same antibody with alveolar epithelial cells on the right versus the skin cells on the left-hand side. And you can see that those different laminins are assembled differently depending on the context. And you get fibrillar rays, and this is 2-dimensional culture in a dish on a glass substrate, so it's a bit different. But when we look at the mice, in terms of the expressing mice, we have this thickened and disrupted with, again, blood passing through it in the alveolar epithelium. big caveat, big flag to wave. These were not inflated at the point of fixation, so it's harder to interpret these data, but it does point that there could be a fibrotic type response going on within our lungs. The liver is not an area that I'm particularly comfortable with, but what we noticed is that the cellularity of the liver was massively reduced. So it's very clear that we have these hemopoietic, we think, colonies of cells in our non-expressing animals that have disappeared, that are much less present in our LaNt expressing animals, and it's quantified down the bottom. So there is an effect on the liver, and I'm more than happy to learn from people who know more about livers than I do. But the last thing I want to show is the kidney. And the kidney, there's two really good reasons. Well, there's lots of good reasons to look at the kidney. It's important. But there's two laminin-related reasons why the kidney matters. So this paper came out in May where this group reported an LN domain mutation in laminin alpha 5 presented with bleeding within the glomerular area and distension of glomerular tubules. Other studies have shown mutations in laminin beta 2 LN domain. So this is S83R, it's just on the edge of the LN domain, and this is past staining, not H&E, lead to glomerular sclerosis. And this is a mimic of a human disease called Pearson syndrome. So LN domain mutations and kidney function are already well established. Okay, so when we look at the kidney, and those people who were here early have already seen this closer up, there's actually a really striking phenotype. We have, actually let me zoom in a bit, we have interstitial bleeding, so you can disagree with any of these points, by the way. This is what we're interpreting. So this red arrow points to regions where we have interstitial bleeding. The black arrow shows regions where we have detachment of the epithelium. And then the white arrow shows dilation of tubules. I'll just show you that zoomed in a bit further. Now, I'd love to tell you that I labeled these things myself. This is what I thought we saw, but we sent it to Rachel Lennon's group, who's one of the groups who works on Alport syndrome and Pearson syndrome, these basement membrane-related diseases. And without telling them what the gene was, they came back with this labeling. So they're talking about a basement membrane disruption along this connection point, which is quite exciting. And there's also, that's their exact words, capillaries in the glomerulus look strange with blood in between them. So we have this pretty striking kidney phenotype. Now at this point, I'd like to show you a whole load of extra stuff, but I can't. The only thing I can show you just now is a pan-laminin stain. And there's no huge disruption. There's potentially thickening in areas, which is probably the opposite of what we expected. But at this point, I have to stop. And the reason I have to stop is because of this bad boy. We hit LaNt Wars episode 5, which is COVID Strikes Back, which means that literally the images you're looking at were the ones that were taken in March, just before we stopped. And so I'm almost going to end. I'm just going to say that the saga will continue. Episode 6 is beginning once again. If anyone has a good title for that, that'd be good. I'll just quickly sort of summarize what I've shown you. So we've identified the missing member of the laminin superfiber family, and we've shown that they've got a wide distribution pattern that means that they could be relevant to a whole bunch of different contexts. And that is something that we have all the tools now to ask those questions. So if there's something in particular that you're interested in, it'd be quite nice to see whether this protein is functional within that region. I've shown you that in certain contexts where remodeling of the basement membrane occurs, we have an upregulation of at least one member of this new sort of subgroup of the superfamily of the LaNt protein. And I've shown you parts of the in vitro functionality data that I've shown before that shows that they have roles in regulating cell to matrix adhesion, laminin organization, and matrix metalloproteinase activity. The first ever in vivo functionality shows that protein is functional in vivo and influences development when expressed early, has pleiotropic effects, affects multiple tissues. And what I haven't shown you today, but I've talked about in the past, is the regulation is actually quite funky. It's got this intron retention alternative polydenylation mechanism that allows for this dual layer feedback at the protein layer, but also inhibition of production. And on that, I'll stop, but I'll just put it as diagrammatic form, just so that we can chat. Oh yeah, I suppose I should, before I do that, let me thank everybody. So all the data you've seen are the people on the left-hand side. Lee did an awful lot of this. The two Valentinas are some of the early stuff in the first part of the mouse. Connor did all the recent transgenic mouse. and Liam has contributed quite a lot to the characterizing the laminin expression. I haven't shown you stuff from the rest of my team. I promise you the next time I give a talk, it's going to be on the guys on the right-hand side. So John has been looking at CTGF, so fibrotic regulation in relation to glaucoma. Thanos has been looking at PAX6, which is the master regulator of eye development, but also regulates the limbal stem cell niche. So he's looking about matrix regulation from a transcript point of view. Jirok is using a laminin fragment to treat burn wounds, and he's got some really nice data that shows that this part of laminin 501 can be used to accelerate wound repair. And Josh is looking at penetration of DHA into the skin. And DHA is the thing that turns your skin brown. So he's looking at self-tan, which is interesting. Along the way, project students and Olivia, who you all know, have involved, and there was a graph from Olivia in here that I skipped past. And of course, a huge thanks to George, Kazoo, Toby, Carl and Hannah and Janet, all of whom have contributed quite a lot of their individual expertise that have, most notably George in making the mouse, Kazoo, MMPs, Toby in breast cancer invasion, Carl and Hannah all the front of the eye stuff, and Janet in the oral SEC, and then my external collaborators who have helped with interpretation and things. And on that, I shall end, and I'll go back to my diagram.
Well, very good, thank you, Kevin. I think I just about managed to keep up with this, and I've heard it before, except for the last bit. So, but I always struggle slightly with some of this because it's so far from my expertise. But you got lots of constant complimentary comments about your photo micrographs, which is always very nice. You have very nice pictures. I think you answered Dharani's question about the human endometrium on your way through. So you have got that. You're just going to, you can contact her offline.
Yeah, I'll dig it out and I'll send it to you because I haven't looked at them at all. So it'd be useful to get some expert opinion on that one. That'd be good.
Rob, do you want to ask your question about bone?
Am I live?
Yeah.
Okay, right. Yeah, so basically I was wondering if we maybe could have a look at the bones as well, because basement membranes are also very important in separating, for instance, osteoclasts from the rest of the environment. Right? Osteoclasts produce acid, hydrochloric acid. It's always been assumed that basically they act under a little canopy of epithelial cells sitting over them. And it's always been assumed that that canopy is very important to keep the rather detrimental bits that the osteoclasts secrete inside. And I don't think anybody's ever really looked properly at what the basement membrane of that little epithelium canopy is. So it would be quite interesting to see if you disrupt the laminin network, whether that affects the osteoclast and bone resorption.
So, I mean, that sounds fantastic. Yeah, absolutely. Let's do it.
Yeah. So one thing that comes with it is, because these animals do have problems with their kidneys, do they survive well? Are they thriving, or do they die quite early? Or
so they were born dead. So this was from the E14.5 induction. All the animals that were expressing were dead. So
basically, to look at adult tissue homeostasis, you'd have to do the Crete treatment in an adult. Yeah. Yes.
But so how do you decide when to give them this, when to put it in? Why 14.5? What was the reason for choosing that?
So we did try a couple of days earlier, and we didn't, and that sort of terminated the pregnancy. It's kind of the earliest point you can do it, and things will still go to term in terms of the tamoxifen treatment, where you can differentiate the tamoxifen from there. The reason we specifically chose that, so this particular model with the ROSA cross with the UBC it's not very subtle, right? It is the kind of the biggest phenotype we'll ever see, and the likelihood is that embryogenesis was the point where we're going to see the biggest phenotype of the biggest phenotype. So in essence, I think the biggest challenge I've had, I think conceptually people are on board that this protein could be important, but we needed something definitive that says, yes, it matters, before we can kind of drill down and begin to ask the sort of finer detail, more refined, more targeted type expression. At least that's the way I've rationalize this story. So the next grant will be lineage specific or temporal, like turning on of the gene in different contexts. And what I'd kind of envisage that being is sort of layering on top of my kind of fundamental thought process here of the different laminin types, and having lineage specific in a 3LN, 2LN, 1LN to ask those sort of paradigm level questions. So not really thinking about this as the LaNt protein per se, but more about the relative role of laminin to laminin interactions in these different contexts. Does that make sense? I went further than your question.
Yes, and if you have this problem, so you started off with the skin diseases, do the people with those also have problems in their kidneys? Or, I mean, because it's involved with so many of the different tissues, how come you get people that survive with just a skin disorder?
So that's where the fact that they have 12, 13 different laminin genes, and the mutation is just in one of them. So they have differential expression profiles. So basically the disease that they get reflects where the mutation is. So, or where that gene is expressed. So it's laminin alpha 3, laminin beta 3, or laminin gamma 2 mutations lead to the skin fragility disorder. Or laminin beta 2 mutations, they have a kidney disorder, or they have, and they have lens defects, malformation of the lens, and subretinal defects. Whereas laminin alpha 2, because there'll be some muscle guys on here, they have a disease called Mearson deficient congenital muscular dystrophy. So they have an age-induced muscular dystrophy because their muscle basement membranes fail. And that's something that we think will happen with these animals once we begin to put them, once we put them under stress, is that they'll get these other diseases too that will actually, because this isn't a genetic disease anymore. We're actually disrupting the basement membrane that's formed, or preventing it from forming, I guess, in this context. There was another bigger answer.
Yeah, it's very good. And anybody else got any other questions? To me, it just raises more questions. Just raises huge numbers of questions.
I hope that's a good thing, because
all research should bring up more questions than answers. But it would be quite nice if you. I guess I find it difficult to understand which bit you address next.
Yeah. So so there's lots of. Did I put that in the end? I was thinking about including that at some point. Maybe I didn't. I guess I could go back. Can I go back all the way? Yeah, so in terms of the big questions here, so we have this distribution and upregulation at various stages. So there's an automatic question there about what's regulating the expression profile, which if once you accept that the protein's functional, then it becomes relevant to ask, why is it expressed where it is, and what controls where it's expressed and when. So this dysregulation in tumor potentially is a target for things. In terms of the in vivo functionality, then the obvious questions are then the more specific ones that we've talked about. And then the in vitro functionality, which I didn't quite show you, what we've raised the questions there, we've actually got quite far down that pathway. But what we still don't know is the kind of, and the cartoon and flip past, the chicken and an egg thing, is we don't know whether it's this causing that or that causing this, basically. So they're kind of obvious progressive type steps. But as you know from speaking to me, I want to ask the big question, which is that sort of the combination of the three, which is we're not quite able to ask that yet. Because I think that this whole area is filled with some beautiful biology. So what I didn't quite sort of emphasize enough is this full length protein, this laminin alpha 3B, is an LN domain containing full size protein. But if we think about the LaNt protein as disrupting anything that contains that, they're produced from the same gene. When the gene is switched one way, it will produce laminin alpha 3B. If it's switched the other, it'll produce the LaNt. And then the LaNt protein will go on to disrupt the laminin. So we have this multi-layered control network of an internal feedback mechanism of a major structural protein that regulates everything. I mean, laminins regulate differentiation of every cell type. If you're not already using laminin in your cell culture to keep your cells happy, then you should be, because it is the number one driver. It's much more important than anything else, as George will tell you.
Kevin, at this point, can I ask a question? I guess what you got from this transgenic is really what you were looking for. I mean, the kidney having the alpha-3 is the most severe. So it is almost a concentration dependent, depending on how much LaNt you are going to produce. And therefore, it reflected where the LaNt is being disruptive, if you like. So maybe in terms of where it is during development, the kidney may be your target, but in postnatal, anything could be a game.
Yeah, absolutely. The only caveat I would say is our kidney staining is in human adults. So it could well be the case that there isn't LaNt in a newborn human or in a younger human. But yeah, I agree with you. I sort of sold it that way, that the kidney was the place to look, because it's already well established that laminin to laminin interactions are really important. So the fact that it came out that way is quite nice, because as you know, we've been looking at lots of things and it's the most striking. Thanks, Rob.
Great. I think we should leave it there, because otherwise we'll just carry on discussing all sorts of things forever and people have other meetings to go to. Yep. Thank you very much, Kevin. Very, very informative, and as other people said, very pretty pictures. I always like that.
Good. Yeah.
Thank you.
Thanks, everyone.
See you all next week.